I'm really happy to have Eran Segal speaking to us today. So Eran just reminded me that I guess I know him since we both started our grad school together. And I thought we started together. Apparently, I started a year before him. But Eran started his grad school studying computer science and machine learning. And did a lot of nice work in Stanford and really core machine learning, mostly on relational probabilistic models, but really a wide range of work in machine learning. And then I was one of the first people to start applying machine learning to bioinformatics and especially to genomics. I think Iran, one of his first papers was one of the first, if not the first paper, analyzing gene expression networks with using tools for machine learning. And since then has done a lot of work on applying techniques to many different areas of biology. Eran is, I think, the only person I know that is in the computer science department. It is a wet lab. I don't know if there's anybody else. Do you know if anybody else? Or
there's probably a few
more. Okay, I don't know them both. And I think really bridges in that way between computer science and not just like applying computer science to biology, but doing real biology. And in particular, has been focusing in recent years on personalized medicine and personalized diet, and using and applying to that a lot of computational tools and tools from machine learning and computer science. So I'm very pleased to hear him today, and I'll teach around.
Okay. Good morning, everybody, and thanks, Nati, for the invitation to visit and speak here, and the opportunity to tell you about our work. We've been very inspired by the progress of what data has brought to the field of biology, and I think the most impressive project that I want to cite is the Human Genome Project that you all know. About 25 years now since the publication of the first draft of the human genome. And that really took us, it's really been a transformative project. It took us from knowing around a handful of genetic variants involved in a few diseases before the publication of the Genome Project to where we are now after genotyping tens of millions of people, where now you could say we know for virtually every disease, hundreds, sometimes even thousands of genetic variants that are involved. And it's also been shown that when you start the drug development process with some genetic evidence for the target that you're after, the chances of success eventually are going to be increased about twofold. So it lowers the cost of drug development by 50%. It's still very prohibitive and in the billions, but it just shows you that this type of data has really been transformative. At the same time, though, our genome is just what we're born with. It doesn't change throughout life. And so it doesn't take into account environmental factors or nutrition, lifestyle, exercise, diet, and so on. And so much like we had the Human Genome Project, we decided a few years ago to start what we call the Human Phenotype Project, where the idea is that in addition to the genome, we want to measure everything else that we can about people, because hopefully by tracking that and also longitudinally, we'll be able to identify health trajectories, predict disease ahead of time, even be able to intervene and prevent it, and hopefully identify also novel biomarkers leading to new drug discoveries. And so a few years ago, we started at the Weizmann Institute in my lab, we started a clinic where we profile individuals. And so before people come in, you can see on the right-hand side, I have this mouse here again. On the right-hand side, before people come in, they upload to a website that we developed all of their electronic health records, and they answer hundreds of questionnaires about medical background and family history of disease and lifestyle and dietary habits and so on. And then they come on site, and on site they do a bunch of body measures. We take blood so that we can standardize blood tests across people, various other physiological tests like hand grip strength, gait analysis, ECG. We record voice that has correlates to disease. A bunch of imaging modalities, so we use a DEXA machine to look at full body composition and bone density, high-resolution images of the retina, ultrasound to look at fat in the liver and the carotids, also for cardiovascular health. We use various wearables, so smartwatches to track physical activity, sleeping devices that with a bunch of sensors on three nights with continuous measurements. We measure sleep at the grade of a sleeping lab, except people do it from the comfort of their home. And we use CGMs, continuous glucose monitors, that track glucose levels every 15 minutes for 2 weeks. During that time, people log their diet. They're incentivized to do that because then they get a report of their CGM coupled with their diet, so they can see which foods spike and which do not spike their glucose levels. They fill in medication they take, any physical activity they do, and we also biobank samples, so live cells, PBMCs, that we can even do functional assays later on, and also serum that we use for a bunch of different tests. And maybe the holy grail is that we couple all of this with the modern multi-omic technologies, and so we also look at the human genome, so that's the genetics, whole genetic sequencing, but then we also look at the oral, vaginal, and gut microbiome. Metabolomics looks at thousands of different small molecules in the blood. You know cholesterol, but we can look at thousands of different small molecules. Proteomics looks at proteins in the blood, RNA sequencing at levels of RNA. We have a novel immune assay that we developed whereby in one experiment, I'll show you this later in the talk, we can actually tell the entire infectious history of a person, all the viruses a person has been infected with and still has antibodies for. So it's a very comprehensive profile, maybe the most deeply, not the largest, but the most deeply phenotyped cohort. We've done this so far on 13,000 people in Israel. It's longitudinal, so every 2 years people undergo all of these different tests. Here's an example of one of these kind of unstructured data that we have. So this is the gait analysis. We actually video people and then we can extract their skeleton at 40 different joints, 60 frames per second, and get recapitulate kind of entire movement of people, which obviously also has correlates to disease. If you kind of bin all of the data that we collect, it covers around 17 different systems of the body. The population that we profile is actually the general population. We never advertise. It's by word of mouth. Actually, there's 30,000 people on the waiting list. We just don't have the capacity to build a large enough lab to capture all this, all the interest. And so we have representatives of 40 different diseases. Of course, we have more frequent are those diseases, the cardiometabolic diseases that are more common in the population in Israel, also in the US population. Then about 30 different medical conditions, so not quite diseases, but still interesting conditions to study, like allergies or vitamin deficiencies. And what's nice is that everybody is measured in the same way on the same instrumentation, so it's very easy to standardize and compare across people. Because we see a lot of value in this data that hopefully I'll show you in the remainder of the talk, all the discoveries we've been making from this. We're now in the process of trying to significantly expand this cohort, with the next milestone hopefully being to reach 100,000 people by both increasing the cohort in Israel, but also by going into additional countries. About 3 months ago, we opened a clinic in Japan. And very soon, hopefully in the coming months, if still in 2025, we'll open another clinic in the UAE, and we're looking at additional countries to open similar clinics in, which will actually make it the first international cohort, definitely on this scale. And I think that's very interesting to tap into ethnic, genetic, and cultural diversity. So compared to other efforts, of course, we're not the only one to look at and build a population-level cohort. Maybe the most impressive parallel study is the one in the UK. It's called the UK Biobank. It started about 18 years ago, 500,000 people, and this has really been a very transformative project. It really transformed the way we do biomedical research because they provide access to the data. There's been over 10,000 publications where you can see the growth kind of being exponential in the past several years, with 40,000 researchers from over 100 countries working on it. Major investments by pharma companies to basically enhance the data set in return for about 6 to 9 months of exclusivity look at the data, and then the data goes into the public domain. So very impressive study. We also use the data. In comparison, obviously we're not as large, but we still have several unique aspects, like as I mentioned, being international, but then also more importantly, all of these more modern state-of-the-art multi-omic technologies like the microbiome RNA sequencing, immune profiling, and metabolomics that we do that are not part of the UK Biobank, and then some additional these more deeply characterized phenotypes like characterizing sleep and CGMs and liver ultrasound. We are also providing access to the data. In fact, if anybody is interested, you can go to this website and obtain access. There's already dozens of academic institutions and researchers working on this data, and hopefully in the coming years we'll start to see publications not just by my group. So far it's been my group, but you can see from the publications that there's been, I think, quite a lot of interest in what can be discovered from this data, and so in the remainder of the talk I want to tell you about a little bit, a few of these discoveries, kind of more from the bird's eye view, so that I can cover a breadth of different topics we've been studying. So one of the earlier works that we've been doing was on trying to use such data to understand what is healthy nutrition, and kind of taking a data-driven approach to the problem. And thinking about this, we decided to look at blood glucose levels. Why blood glucose levels? Because it's maybe the only measure that is quantitative that we can look at in response to single meals, as opposed, for example, to looking at changes in body weight in response to a diet, which, as you know, takes weeks, if not months, to observe a change. And so what we did was we connected people to these continuous glucose monitors. They ate their habitual diets, and they logged and we recorded what they eat, but also in the morning we actually provided the breakfast. And this is an example of 4 out of, in this case, 1,000 people that were profiled, just to illustrate a phenomenon that we saw on the scale of 1,000 people, that when you have different people eating exactly the same food, we saw very different blood glucose responses. So what you're seeing here is in the 2 hours after eating a meal, the changes in the blood glucose levels. Different colors represent different people. The same color is the same individual on 2 different mornings. And so you can see very reproducible responses within the same person, in this case, when eating 4 slices of bread in the morning, but very different responses between people. And so this tells you, without even telling you how we addressed the problem, that if you want to balance blood sugar levels in individuals, which we know is critical for weight management and other diseases, the solution has to be personalized because people have very different responses. So it means that if you want to solve this problem, you have to collect some personal data about people, which is what we did. And at the time, we focused on various clinical parameters like body measures, blood tests, and also features having to do with the microbiome, which we know is very much involved in our metabolism of food. And we were able to develop a machine learning algorithm that predicted, not perfectly, but quite accurately, the blood glucose response of individuals to single meals. So here, every dot is 1 meal, and it's quantified by the area under the glucose curve in the 2 hours after eating a meal, either what is measured on the y-axis or what is predicted on the x-axis. And you can see a fairly good correspondence, such that we could even take people who are pre-diabetic, where for 1 week of CGM data, you can see many spikes in glucose levels, reaching also abnormally high glucose levels, indicating that this participant here is pre-diabetic. And then for the same participant, just 1 week later, also connecting this participant to a CGM and prescribing a diet based on the algorithm with exactly the same amount of calories in every single meal, we could fully balance blood sugar levels in this individual. So this we published kind of the idea and a simple intervention of 2 weeks, and then we decided to do a full-blown RCT, randomized clinical trial, on 200 people with prediabetes to see the long-term effects of following such an algorithm-based diet. So we randomized 200 people into either a standard of care Mediterranean diet or the algorithm diet. And here, anecdotally, if you look at one participant, this is 1 month of CGM data, where you can see on the habitual diet prior to the intervention, many spikes in glucose levels, and then in the 1 month beginning of the intervention on the algorithm diet, we see pretty much full normalization of blood glucose levels. And then statistically, when we look at the result of this RCT, if you focus on the center panel here, this is the changes in hemoglobin A1C, which is the gold standard for diabetes. It's a blood marker that measures your average glucose levels in the past 3 months. You can see in red on the Mediterranean diet, after half a year of intervention, a significant reduction in hemoglobin A1C as would be expected, but double the effect on the algorithm-based diet, and even more significant effects if we look at various CGM parameters like changes in various spikes in response to meals, and also improvement in several other metabolic parameters. So this for us was actually a journey of almost 10 years, going from an idea to developing an algorithm for predicting blood glucose response to food, to actually doing a full-blown randomized clinical trial, which we view as a really treatment for people with prediabetes. And we had a follow-up paper showing that this is also effective for people with type 2 diabetes. So very gratifying going kind of from an idea to actually testing this in real human subjects and showing that it's effective and could be used as a treatment. Continuing on the thread of the microbiome, we have a major interest in understanding the influence of the microbiome on our health status. Yes, please.
So in the previous study, I have 2 questions. So
Please.
these test subjects, were they part of the training set also, or you either clustered like people in your training set to give them personalized recommendations? Do you do all these diagnoses for the people in the test set? The second question is, how would this compare to a baseline where a human dietitian or physician had access to the medical tests and were giving a personalized diet?
Yeah, good question. So first, when we went into this RCT, the 200 people with prediabetes, we used an algorithm that was actually developed prior to that on 1,000 different subjects. So you could consider all the 200 subjects as being kind of a test set that we collected data from them, but we did not use that data to retrain the model. In terms of comparison, the comparison that we did here was actually to the what is the standard of care. So this is actually what dietitians, even to this day, prescribe to people with prediabetes or type 2 diabetes if they want to do some dietary intervention, which is a Mediterranean diet. It was also tailored to individuals because of various food preferences or lifestyle, access to food, and so on. But it kind of followed the principles of a Mediterranean diet, which is now the standard of care. So when you typically propose a new treatment modality, you need to compare it to the standard of care, which is what we do. Yeah, and actually feel free to ask questions during the talk. It'll make it more interesting. Okay, so continuing on the thread of the microbiome, we really have a major interest in this, both looking at the relationship between the microbiome and our health, but also looking just at the evolution of the microbiome itself. It's an ecosystem within each of us. Actually, the number of bacteria you have in your body equals the number of human cells that you have. They're just smaller, but in number they're bigger. They actually have 100 times more genes, millions of bacterial genes within each of us compared to about 25,000 human genes. And so one of the studies that we did was to study the relationship of the microbiome and human health. Actually looked at the microbiome at a much deeper, higher resolution than what is typically done. And so typically people just look at the composition. By composition I mean they look at the types of bacterial species that we have and correlate that to various health status. What we did here was actually follow a very similar approach to how we study human genetics, which is to look at single positions within the human genome and look at variations. It's called single nucleotide polymorphisms, and associate these SNPs with human traits. So here we did the same, except that we looked at single positions within individual bacterial species and associated changes in genetic variation in individual positions with a human trait. So here's a slide that illustrates the concept. We take a stool sample from people, we sequence it, most of the DNA is going to be from bacteria. We map it to a reference bacterial genome. So here's an illustration of one species, another species, where we map the individual sequencing reads, and then we can record the genetic variation that we see at each position in every bacteria. And so in this illustration, you can see at this highlighted position, we have an A for the first individual, a T for the two other individuals, and a higher BMI for the first individual compared to a lower BMI for the other two. So you would consider this to be an association between genetic variation at this position and a human trait. But of course, this is just an illustration, and we do this on the scale of all of the data. When we did this analysis, we froze the data set at about 7,000 samples. And this is a standard, it's called a Manhattan plot, standard for human genetics, except that in human genetics, you typically have human chromosomes on the x-axis. Here we have bacterial species. Every dot here is one position. It's a SNP, where we show here on the y-axis the significance of the statistical association between genetic variation at this position and a human trait, in this case, a BMI. And you can see above the red line, which controls for multiple hypotheses, we see many SNPs that are coming up at very high significance levels. Please, question.
What kind of bacteria? What's specific? What kind?
So we actually look at all the bacteria that we have in our gut. We look at the gut microbiome. There's about 3,500 different types of bacterial species in total in the human population. And each of us has on the order of 200, sometimes even 500 different bacterial species. So we take all of the data and we map them to these reference genomes. It's about 3,500 different bacteria, and then we record at each position the
It's well known that there are healthy bacteria and harmful bacteria, so it depends on the person.
Right. So these are the commensal bacteria. So these are bacteria that are in general found, as I said, there's about 3,500 different bacteria found in the human body. I think good bacteria and bad bacteria is a simplification, I would say. You can have the same bacteria, the same bacterial species, depending maybe even on the exact strain and mutation. In some contexts, it could be a beneficial bacteria. In other contexts, it could be a harmful bacteria, and also it could change its context depending on other bacteria that are also with it in the ecosystem.
Healthy human, or they have diarrhea or whatever diseases.
Yeah. These are generally, I mean, this is the population I showed before, so I would say generally healthy people. Some have some diseases, but here we looked at the trait of just BMI.
How homogeneous is the genetically homogeneous of the bacteria inside each individual? Because I mean, the way you're describing it here, you're saying you take a species and you look at its variation.
Yeah. So that's a good question. So when I said before that there's 3,500 bacterial species, the definition is when two genomes are further away than 5% of their genome, then they would be considered different species. So there's 3,500 different species, meaning that let's say we're talking about E. coli. So all of the E. coli that we would all have, because we're still considering them the same species, their genome would be identical to 95% and higher between two different individuals. Typically, it'll be around 98 to 99% identity. which actually shows you that it's actually quite a lot of variation because every two human individuals are roughly 99.9% similar.
No, but what I meant is the variations you're measuring. Like, if are all for the E. coli, are all the E. coli in my body have the same genetic makeup, or there's less variability inside that? Because when you're measuring which which SNP do I have, right?
Yeah.
I mean, my genome has one
Yes.
SNP or two.
Yes.
I don't know, but here.
Yeah, that's another aspect, right? So first of all, so your E. coli and my E. coli would be, again, just because of the definition, they would be very similar to 98 to 99% similarity. But then you're right that also within the same person, we can actually sometimes have multiple different strains. So actually, when we record this genetic variation, I simplified here to say we have an A, but in reality, we can have multiple different reads coming actually from different strains, and what we would then actually compute is the allele frequency of every of say A and T, and maybe in U there's this A is present in 95% and 5% is a T. So we would actually we actually quantify that number, and the association that we do is between that allele frequency and the trait. In most cases, there's actually typically it's one SNP per person at different positions, because locally within the same ecosystem, there's typically would be an advantage to one strain over the other, and it would overtake the population. But we sometimes do see multiple different strains within a person. So yeah, good point. So anyway, we have many significant associations, which is actually very interesting, because if you think about it, we're talking about just one position in one specific bacteria that shows very significant associations with a very complex trait like BMI. Even more interesting than that, though, is if we plot the same data as a volcano plot, where the y-axis is, again, the minus log p-value, but the x-axis now is the number of BMI points that are statistically explained by this variation. So in other words, the effect size, you can see that we have multiple SNPs that actually achieve a very high significance level, so explaining 1, sometimes even 2 points of BMI. Here's an example. So here we partition the individuals into 3 groups for this particular bacteria. There's some individuals that just don't have this bacteria. Other individuals that at this one particular position, they have this bacteria and they have the major allele. That's 2,000 people. And then 400 individuals that at this position, they just have a different allele, they have the minor allele. And just by separating the individuals based on this one position in one bacteria, we can see that there's a difference of almost 2 points of BMI. This can be 20 pounds of body weight. It's a huge effect. It's actually bigger than the largest effects ever seen for the human genome, which is very interesting. We had earlier work that showed that there's very loose connections between the human genome and the microbiome. In other words, the microbiome is mostly an environmental factor. It's not influenced so much by our genetics. And so to me, it makes a lot of sense, because by being an environmental factor and having a connection to BMI, which we know is very much influenced by environment, there's also a genetic component, but very much influenced by the environment, it makes sense that there would be a microbiome connection. But nevertheless, the results are very strong and striking. And so what we are doing now is we're now trying to go back to individuals who are lean and have the variants that at least are associated with lower BMI, and trying to prove causality, so trying to extract the strain from these lean individuals and give them in randomized clinical trials to people who are overweight and see whether we can have a positive beneficial effect on weight loss. And similar to doing this on BMI, we do this now for many different traits, and we find actually very significant hits and very large effect sizes for many other indications.
But BMI, so you have like obese people, overweight people, and you have so how do you like? Is it related to their composition, to their body composition, or how, like, can you a little bit more elaborate on this?
So we try to look at to try to understand the mechanism maybe behind this, but I have to say this is proven quite challenging. So sometimes we would, in some cases, we could have hypotheses. We would see that there's a mutation in a gene involved in energy metabolism, say, and then we can hypothesize, okay, maybe that's two individuals eating the same food. One could actually extract more energy from the same food compared to the other, and maybe that's a mechanism. But in other cases, we see an association and we don't have a clear connection to the gene in which we see the mutation and the trait of BMI. This actually is very similar also in human genetics. We find many hits genetically to many traits, and in many cases it's very hard to understand the mechanism.
This cohort was otherwise healthy, or what they?
Yeah, these are mostly healthy people.
Healthy?
Yeah. Okay, I mentioned the profiling of the immune system. So here I won't go into all the details, but we have a high throughput way by which we actually can synthesize hundreds of thousands of antigens and incubate them with antibodies from the blood of a person, and then measure all of the antibodies, all of the antigens that are bound by a person's antibodies. And so because we have the capacity to look at hundreds of thousands of antigens, we can actually print and measure all of the different viruses. And when we do that, we see that the immune profile of individuals is actually such that on average a person recognizes about 1,000 antigens. There are some that are very common, so recognized by many people. There's viruses that infect the majority of us, and then there are others that we would consider as being more private. So many that actually in just one individual we find a hit. In general, this immune marker is personalized. So if you look at the correlation of this immune profile within the same individual across different years, we see a very high correlation, much higher than just a correlation between two random individuals, which are also to some degree correlated because we are exposed to similar viruses, but it really is a personalized fingerprint. And so we use this to study various disease cohorts, like this one on chronic fatigue syndrome, where we looked at matched healthy controls to 40 chronic fatigue syndrome patients. And in comparing them, we could identify many different antigens that are much more enhanced and bound in chronic fatigue syndrome patients, coming from flagellins. So this is the part of the bacteria that is the motor of the bacteria. So we hypothesize here that maybe some mechanism is that there's been more motility of bacteria within chronic fatigue syndrome patients that perhaps causes inflammation or other aspects of the disease. But in any case, we can then take this data and develop a simple machine learning algorithm that predicts with fairly good accuracy and distinguishes healthy from chronic fatigue syndrome patients. And this is important because until now there's really no biomarker for this disease, and we found one in the immune system. Another type of data we look at is what's called metabolomics. So this is the set of small molecules that we can measure in the blood. And so a few years ago we did this very, I would say, fundamental work where we looked at all the different small molecules, these metabolites that we can measure. And because we have different sources of data, like the genetics of a person, the diet of a person, their microbiome, and so on, we could ask the question of what explains statistically, again, the levels of different metabolites. And we found that some metabolites are well explained by diet, others by microbiome, and others genetically. And so this was kind of a fundamental database because, and it's important because if you think about small molecules that circulate in our bloodstream, we know that they're important for our health. And once we identify one that's important in some disease, the next thing that you'll want to do is to modulate that, and to modulate it, you need to know what's the determining factor. And so using this database, we could then shed light in certain disease cohorts. So for example, This is a study we did on people with who underwent a heart attack, acute coronary syndrome patients, and we showed that you can take even just a single patient and extract a healthy cohort that's personalized for this patient. So let's say we have a 60-year-old male who underwent a heart attack. We would take 60-year-old males who are healthy, and we can then look at each metabolite and build a distribution of what would be the expected normal levels of this metabolite that are the reference for this patient, and then see where the levels of this metabolite falls for our individual patient. Here, it's actually outside of the distribution. We can compute the p-value, the number of standard deviations away from this expected distribution. We can do this for all metabolites and arrive at a metabolite deviation profile for this one patient, and then we can cross it with the database of determining factors for each metabolite to ask whether the deviations that we see in the metabolite profile of a person originate from metabolites that are mostly linked to diet or genetics or lifestyle factors and so on and so forth. And when we do that, we see that even if we have two, say, 60-year-old males presenting at the clinic with the same condition of a heart attack, when we analyze their metabolite profile, we can sometimes see that the metabolite deviations are very different from these individuals, where in one individual they could be mostly driven by diet and another by microbiome, and that may tell you something about the pathophysiology of disease, which may be different, and therefore also the treatment should be different between these individuals. Another major interest of ours is in aging, and this is actually a nice example where we can utilize the many different data modalities that we have. And so here we took one of our data sets, the, again, metabolomic data, and we used it to predict the actual age, the chronological age of a person. And this is the result of such a machine learning algorithm. Every dot is a person. The x-axis is the actual chronological age of that person. The y-axis is what is predicted by a model that only looks at the metabolite profile of a person. And you can see we get fairly good predictions, but not perfect, because individuals here at the top, when the model looks at their data, they look more like somebody who's older, and conversely, people at the bottom, when the model looks at their data, they look like somebody who's younger. And indeed, we saw that if you compare these individuals who are predicted by the model to have a higher chronological age than they do compared to individuals who are predicted by the model to have a younger chronological age, we see that individuals with a higher predicted age are indeed in worse clinical condition. They have higher BMI, triglycerides, hemoglobin A1C, blood pressure, cholesterol, and liver enzymes. So they're in worse clinical condition. And so we call this kind of the biological age in contrast to the chronological age of a person. And we can do this for different modalities. So this is for the RNA data. And again, we see that individuals predicted to have a higher age are in worse clinical condition. We can do it for the other modalities like the retina scans. And in general, we find a quite surprising result, which is that if we look at these different body systems and different, we call these biological clocks for different body systems, and we look at the correlation between these different clocks, then by and large, you can see this cross-correlation matrix is rather sparse, indicating that that we see pretty much independence between the clocks. So if you're aging faster in one system of the body, it doesn't necessarily mean that you're aging faster in another system of the body, which is actually a result that surprised me. The second thing that we see is that these clocks are biologically meaningful. So if you have a higher biological age, you typically are at higher risk of developing a disease related to the body organ on which the clock was developed. And finally, we see a difference between males and females, with males aging pretty much linearly, and in females we see this big jump between the age of 50 and 55, which we could actually link to the menopausal status of the women, because comparing women of the same chronological age, post versus pre-menopausal women, we find that post-menopausal women have higher biological age on several of these different biological clock. So interesting.
So I'm a bit confused. If you go back to the previous but what's the? I mean, in some sense, what's the y-axis here? Like, I mean, they can apply some monotone transformation of it. I mean, you call it age, but if it doesn't actually correspond to age, as we see in the right plot here, is it because it's normalized for both genders, or why? I mean, I don't understand how you can get something like a plot there, right? Because what's the units in the y-axis?
So the units of the y-axis are actually chronological age.
So why doesn't it correspond to actual? But it doesn't actually correspond even on average to chronological age.
So you can see that it actually does correspond, right? I mean, this is just an algorithm that tries to predict, and you can see here that the predictions they look qualitatively different between males and females. So for females, right, you don't get these. The predictions don't make this kind of linear line. It's just that.
I mean, is it calibrated in women? I guess.
If the model is calibrated at the end.
Is this model on women actually calibrated?
This is just the result of an XGBoost model of using the blood metabolites in this case to predict to regress against chronological age, and this is what.
Okay.
So these all facts are well known in medicine. It's like what you made another perspective on in this. What is a novelty? What is a novelty?
So as far as I know, actually, this type of result is quite novel. That we look at, we can identify specific body systems where, if you compare women of the same actual age, chronological age, those who are postmenopausal actually have a higher, we call this biological age on these different body systems. And this, as far as I know, was not reported. And then finally, with age, we see that these models are also with prognostic value. So if we take individuals at baseline and we compare what happens to them in the future, then people who are biologically older at baseline, if we look 4 years into the future, they actually are more likely to develop, in this case, cardiovascular disease, and then as compared to people who have a younger biological age, so it also has some prognostic value. And then finally, in the last few minutes, I want to tell you about the kind of what we're mostly concentrating on right now, which is applying the more, let's call them next generation types of AI models, mostly transformer based models to these data, where the goal is to be able to hopefully integrate different modalities into a more holistic view of human health of a person, because we view each modality as giving us a different window into a person's health, looking at different aspects. And on the path to doing that, we're looking at one, sometimes two modalities and building these models. So one of the more successful ones we were able to build is one based on the glucose levels. So here we took a pretty standard transformer-based model approach to the way that you learn large language models by predicting the next word. We built models to predict the next glucose level. And so then we have a generative AI model for glucose levels, which can view the history of some historical data of a person, let's say within a day or within several days of CGM, and then generate plausible trajectories, plausible completions of the CGM data that look pretty similar to what the real CGM data looks like. But more interesting than that is actually, much like in LLMs, we're not just interested in predicting the next word, we're interested in the underlying representations and what we can apply them to. So here too, we showed that the representations are actually quite useful, because we can take people who are pre-diabetic, so their hemoglobin A1C is within the range of 5.7 to 6.4. And quite surprisingly, we see that if you partition these individuals into kind of the lower end of hemoglobin A1C versus the higher end within the range of prediabetes, you see that people at the higher end are actually not more likely to develop diabetes in the future. But if we take the representations of the CGM model, transformer model that we built, and we partition individuals, then we begin to see a significant separation in terms of who will develop diabetes 2 years into the future. And we also validated this on an independent cohort from Spain that we did not use for training, where again we see that hemoglobin A1C itself is not predictive of who will develop 10 years into the future diabetes, but we call this the gluformer model for glucose and transformer. We see it actually is predictive of who will and who will not develop diabetes 10 years into the future. And so we have a bunch of these different models. Maybe I'll skip some of them, but from the microbiome, from the ECG. The ECG is interesting in the sense that we train it with a different type of approach, with contrastive learning, where we try to bring together the alignments of the model if the ECG signal is coming from the same person, say the first 5 seconds versus the last 5 seconds of the recording, or if it comes from the same person separated by 2 years, and we try to push apart the embedding vectors if they come from different people, and we learn useful representations of that. Another model was targeted at cardiovascular health, so linking together two of the different modalities that look at and are relevant for cardiovascular health, the high-resolution images of the retina, where we can see the blood vessels non-invasively, and the ultrasound of the carotids, which also is informative of cardiovascular health. And here too, we use this contrastive learning framework to kind of pull together embedding vectors if they come from the same person, let's say the left eye or the right eye from the retina, or the carotids and the retina if they come from the same person, and again push apart embedding vectors of different people, and also show that we can learn a useful representation. And then finally, the model that we actually are working on now, we call it HealthFormer, where here really the idea is to bring together all the different modalities, and you could imagine many different approaches to doing that. One that we are using is to actually take all of the 30 or 40 different modalities that we have on a person and put them on a timeline. So you can think of different medical events that we have for a person. So every time we measure a blood test, or we have a drug that somebody is taking, or a diagnosis, or a microbiome test, and so on and so forth, we can just put them on a timeline. And then we can also use this framework of next token prediction to take the historical data that I saw for a person and then try to predict the next medical event. There's a few technical challenges here because we have different types of modalities, and also because time is irregular. Sometimes when I have data during the night and I measure the sleep of a person, I actually have several measurements per second. And then other times a person would visit the clinic and then visit only 2 years later. So now we have a gap of 2 years. So there's very different time scales that we also have to deal with within the same model. So there's a few challenges, but there's already some interesting preliminary results. But I would say when we consider the data in this way, then we actually have about 20 million, depends how you count, but about 20 million data points per person times 13,000 people or 20,000 visits. That's hundreds of billions of data points. Of course, it's not balanced. So some modalities like sleep, that we have multiple measurements per second, have a lot more data than, say, blood tests, where we have dozens of measurements over time. But still, it's a lot of data that we can predict. And hopefully, if this is successful, then I think it can be quite a useful model, because it can act as a digital twin of a person, whereby we can then also predict health trajectories, including your health trajectory under certain interventions. So you could just, if you have such a model, you can just inject synthetic tokens mimicking different interventions like a change in lifestyle or medication and so on and so forth, and use the model to predict what would be different health outcomes, and by that also be able to recommend different actions to improve one's health. And so in closing, I would say we believe there's a lot of value in such data, which is why we're now also working very hard to try and scale up the project to reach 100,000 people. We also want to measure more phenotypes. There's very comprehensive measures that we're doing so far, but obviously we're not looking at everything that can be measured. We want to capture more diseases, look at additional countries, as I mentioned, to tap into cultural and ethnic diversity. And I really think that this is the type of data that can really be used, especially now with the new AI models, to really build the next generation of tools in precision medicine. And with that, I will stop and happy to answer any more questions. Thank you.
So back to when you were talking about BMI and the gut microbiome, you were asking about looking at the causality. Is it possible that it's kind of more complex, like both directions, some sort of equilibrium where if BMI gets larger, it makes the body more hospitable to this kind of microbiome, that kind of microbiome, bacteria then makes it, changes metabolism, and then things kind of go both ways, and sort of it sits like that. Or I don't know.
Possible. I mean, I think traits like BMI are very, very complex, and probably there's, and obviously, but microbiome is not the only determining factor if it is all determining. So, I would imagine many different complex interactions. I will say, though, that because we are looking here not just at which bacteria you have, but actually at the genetics of the bacteria, for me, it's actually very hard to imagine that a trait like BMI would induce such selective pressure to the degree that it would actually influence individual nucleotides in specific bacteria. So I actually, of course, we have to prove this, which is what we're trying to do, but I actually would think that the causality is actually working in the direction of, your microbiome, which we know is very much involved in the metabolism, is giving you some propensity for, in this case, obesity. And then it could be realized depending on what you eat and lifestyle and so on. But of course, unlike genetics, where of course we know the causality is going just one direction, here we don't know that formally, so we have to prove it. We have to do the experiments, and it's complex and hard, but we're going to try to do that.
Two questions. One is, so I'm curious what your vision is for the future in this. Like you're doing now these tests with people. I'm curious, like how much does it cost per person? And in order to use this for actual medicine, do you expect that we will routinely have this type of test that you do now, and feed that into your transformer and get our recommendations, or is this data only necessary for research? But then when you actually use it, you'd use different types of data.
So first of all, yes, this is a very expensive project. Although we did spend and are spending a lot of effort to reduce costs by high throughput. And robotics and doing the multi-omics in that way. It's still very expensive, probably in the several thousands per person, which is still it's quite cheap.
Wait, that's not expensive.
Well, it's quite cheap compared to what we measure, but it's very expensive when you multiply that by 20,000 visits that we had so far, and by
Wait, this is per visit or per patient?
Per visit. But okay, so even if you have, let's say, 5 visits, so over the course of 10 years. So I would say, but it's still, it's cheap compared to what we measure, but it's expensive if you think of scaling this up to a population level. And so I hope that, or I would say, yes, the vision is to build this data set as large as we can, but use it to build these models and then be able to reduce the number of tests that we do per person when applying it to actually make predictions. and hopefully also use, and we're developing some of these models to kind of impute one data set, to impute an expensive data set from a more cheaper and easy to obtain data set. So I think this is where we'll try to head to be able to scale this up to a population level.
The other question, a bit more technical and related, also I guess somewhat to what everyone asked, what you mentioned that you have different timescales. Do you try to explicitly model latent latent variables of what's going on there, like between visits in somewhat the same way that with transformers, and we do use actually latent generation for chain of thought.
Yes, so we don't do chain of thought right now,
Well,
but we
not
do.
exactly chain of thought here, but like
But the models are we are building these transformer and very large networks. So you could say that they have a lot of latent variables, right? That many of them we actually don't understand exactly, what they're trying to do. But they do capture in a latent space information that allows you to generate these predictions. Okay.
Okay.
I have two questions, but I know we're short on time, so maybe you can choose which one sounds more useful. So one is maybe a follow-up to Nadi's first question, which is when you learn these models to predict something, future glucose or whatever, presumably you're inputting lots of features, some of which are important, some of which are not. Can you already interpret some of these results to say, oh, actually, this is the one additional thing we should be measuring that we're not? That's one question. And the second is when you're talking about the number of tokens of different modalities in the language models, I was just curious, what is a token, and have you looked at different tokenizations for language models? There are different possible tokenizations. Yeah.
Right. Let me try to quickly answer both of them. So first of all, yes, once we learn these models, we go and try to introspect them, with the standard tools like ablation studies and so on to see which features are actually driving the predictions. And beyond the obvious, for example, like your cholesterol level in 2 years, my first prediction would be that it would be similar to what I measured 2 years ago. But then beyond that, what helps me generate the predictions, especially which other type of modality. So we try to look at that. And then in terms of the tokens, right now, of course, there could be many different approaches to building these models and also to doing the tokenization. Right now, we kind of consider every type of measurement as its own modality. Okay, so if we have multiple different blood tests, each blood test is a modality, and the different tokens that we generate for it are actually some quantization of the space. So we look at, say, depending on how much data we have, 10, 20, 50 different quantiles from the data, and each of those becomes a token.
So like, for example, for sleep, which you said is measured every second, whereas some other things are not, I could imagine, an hour's worth of sleep data being a token.
Right.
How do you decide on that?
So we look at it much more atomically. So basically every measure that we have, right now our tokens are mapped pretty much one-to-one to every physical measure that we do. So if we measure, if we have a measure done at 60 hertz, 60 times per second, then we would actually have, each of them would be a data point, and we would tokenize it into, as I said, let's say 20 different methods. But you're right that there could be other approaches, and we could go to a higher level and build tokens on a more complex, like a pattern of some time series, which would be a token. We haven't done that.
So you had a spider plot where you were measuring some variable and you were saying you identified two types of effect, microbiome and diet. I was slightly confused because both of them seem extremely correlated. Isn't diet the main environment factor that would contribute to microbiome, or?
So in that particular plot, we were looking at kind of the metabolite deviations of different people, and we were looking at which factors are determining these different metabolites. And so in that case, there were some similarities, but some differences. So in one person, there was a bigger contribution to microbiome-driven metabolites and another more to diet-driven metabolites. But there was also some degree of correlation because in both of them, both factors contributed.
And there's a question here. Okay, so there's more questions. Let's thank you again.
Thank you.